now that you understand what Darwin's theory of natural selection actually is, you're probably saying to yourself, okay, well, how can we know that this is true? And so this last section in chapter 19 is about the evidence that we've found for evolution. There are basically four types of data that we have as evidence that evolution has occurred. The first being direct observations of the world around us. Second is homology, and I'll explain what that is. Third would be the fossil record, and then finally, biogeography. So, direct observations of evolutionary change include examples we have actually witnessed of natural selection, and I mentioned in the last lecture about how it's really hard to observe natural selection in humans because of our long lifespan. But in some other organisms that have shorter lifespans, we can actually observe this happening in our own lifetime. So one example are the soapberry bugs, and these are bugs that use a little beak they have to feed on seeds. So you can see here's a soapberry bug right here. They have a little beak. And they usually can eat the best when their beak length is similar to the depth of the seeds in the fruit. So if you have to go far into the fruit, a longer beak is better. If you have to go less deep into the fruit, a shorter beak is better. So in southern Florida, where there's soapberry bugs that eat larger fruit from balloon vines, those soapberry bugs in southern Florida have longer beaks. But in central Florida, we've noticed they feed on a smaller fruit, and those have shorter beaks. So these graphs kind of show the beak length, and in central Florida, those goldenrod trees have shorter beaks, and then on the southern Florida, the long balloon vines, they have longer beaks. So they've evolved to get the seeds in the fruit that is most common where they live. In all cases, the beak size evolved in populations that feed on plants that were introduced with fruits that were smaller or larger than the native fruit. So when new plants were brought to Florida that had different size fruits than what was originally there, the soapberry bugs evolved. And because of this introduced species, we were able to observe this quick evolution in beak size in less than 35 years, whereas we talked about a lot of times evolution takes a very long time. Something else we've observed, which is a little bit frightening, is the evolution of drug-resistant bacteria. So you probably are familiar with antibiotics, which are usually used to help get rid of bacteria. And the bacteria Staphylococcus aureus is often found on people's skins or nasal passages. So if you've ever heard of someone having a staph infection, that's probably caused by this bacteria. However, there are certain strains of staph that are methicillin resistant. This is an antibiotic, methicillin, and so we call these MRSA, basically representing the acronym of this. And these are very dangerous pathogens because they are resistant to most bacteria. Because staph, once we started using penicillin, the staph bacteria became resistant to that. Then it became resistant to methicillin. And so these are very dangerous because we don't have a lot of antibiotics that can take down these staph. The way methicillin works is by inhibiting a protein that is used by bacteria in their cell walls. But MRSA bacteria use a different protein. So when exposed to methicillin, the antibiotic, it kills all the bacteria that have the protein that it works on, but the MRSA strains that are resistant are more likely to survive and reproduce, and now they're going to become the more common strain in the population, and therefore that bacteria becomes resistant to our existing antibiotics. So this is why it's really important not to use antibiotics if you don't need them, because it propagates the reproduction of those bacteria that are antibiotic resistant. So you can see these are the hospital admissions for people with methicillin-resistant staph, and you can see they just continue to grow because over time, those bacteria that have the methicillin resistance continue to survive and reproduce. Once again, this is a reminder that natural selection doesn't create new traits, but selects on traits that are already present in the population. And the environment that those organisms are in will determine what traits are selected for or against. The second piece of evidence is known as homology. So this is the homo, is a root that means similar. So homology is basically where we observe similar characteristics in related species that may have different functions, but we know that their similarity results from a common ancestor. So homologous structures in organisms are anatomical structures that are very similar that represent a theme present in a common ancestor. So the most common example used for homology is the arm and hand bones. You can see in a human, a cat, a whale, and a bat, they all have similar bone structure, but they use these appendages for different things. Bats use them for flying, whales use them for swimming as flippers, cats walk on fours, humans use their arms differently, but they all have very similar bone structure. That's what we call homologous structures. We can also use comparative embryology to see anatomical homologies that aren't visible in adults. So if you compare a chick embryo with a human embryo, you can see both of them have these pharyngeal arches that are kind of like gills. Both have a tail. Obviously, a human, once they are born, looks very different than a chick, but at the very basic level during development, they look almost identical. You might not even be able to tell the difference between them. Vestigial structures are remnants of features that perhaps served an important function in the organism's ancestors but are no longer used. For example, our appendix. We no longer use the appendix, but it's still present, so it's a suggestion that perhaps it was there in the past. Whales also have a pelvic bone that they no longer use, but is present in them as well. In addition to homologous structures, we also have an example of something called convergent evolution, and these are what are called analogous structures. So this is convergent evolution is when similar structures arise in distantly related groups. This is usually due to the fact that these organisms live in similar environments, and so they adapted similar traits. But you kind of have to be careful when you're looking at these traits, because sometimes traits are homologous and organisms do share a common ancestor, but a lot of times it just happens that they evolved in similar environments. So, for example, the sugar glider found in Australia and the flying squirrel, which are found in North America, both have this skin structure that allows them to kind of glide in the air and go from place to place. You would think that these organisms would be related, especially based on the look of them, but we do not currently believe that they are related to a close common ancestor. We think instead they evolved these features separately, but the south coast of North America and the area of Australia have very similar environments, very similar food sources, and so we think that they evolved in these separate locations totally separately, and this would be an example of convergent evolution. The fossil record also provides us a lot of evidence of evolution. In the fossil record, we can observe extinction of species, we can observe how new groups originated, and also changes within groups over time. So for example, we can see how so many different mammals have toe-like structures. So here's a dog, a pig, and a deer, and then this is a now extinct organism, but they all have toes. Fossils can also document transitions in organisms. So for example, movement from the land to the sea. We can see how we moved from, say, whales to these other organisms, and that ended up developing legs and were eventually able to walk. But noting that they all have similar bones observed in their fossils. Fossil evidence also shows that these living cetaceans, like whales and their close relatives, the even-toed ungulates, are more different from each other today than were the early types. So the early ones were very similar, whereas now we see there's a strong difference between whales and deers and hippos, etc. But at one point, they were very closely related. We can also study the biogeography, basically where different species are located on the planet Earth. This also provides evidence of evolution. We believe that at one point all the continents were united in a single large continent called Pangaea. And so organisms were probably able to move freely between continent to continent. But then over time, the continents separated out as how they are now due to the shifting of tectonic plates and whatnot. And because we see fossil evidence of similar species on different continents, we can kind of see how the Earth may have changed and shifted over time, but also how species would have changed and shifted over time in response. In science, a lot of people will try to argue that Darwin's theory is just that, just a theory. And so it's important to kind of note why we do accept evolution as the prominent theory in the changing of organisms. So a theory means that we have many observations and explanations and evidence for that theory, but we also are still continuing to test and add to the theory. So it's not completely finished yet. So it's not that it's incorrect or wrong or that it's up for debate. We do believe that organisms have evolved and changed over time according to natural selection, but there are still many things that we're trying to understand and figure out about natural selection, and so that's why it's considered a theory, not a law. Basically, we form a theory we begin by forming observations, and then we make inferences, and then we try to find evidence that supports those inferences. So that is the end of our lecture on evidence on evolution.